Hey everyone, welcome back to Foundations of Immunology for MLTK 2700. And I am Sarah Steinberg, the instructor. All right, so in part one, we started building on the foundation of the immune system. So now we're going to start putting those pieces together. This lecture is really about the two big arms of the immune response, the innate immunity and the adaptive immunity. Instead of thinking about these as two completely separate systems, just like we discussed in part one, I want you to think about them just as different parts of the same response. One responds quickly, the other takes a little bit more time, but it's much more specific and can actually remember what it has encountered. And importantly, they communicate with each other, right? So as we go through this, don't get too caught up in trying to memorize every individual detail. Keep asking yourself who responds, what are they responding to, and what happens next. And as we go through this lecture, I want you to continue to think about the imaginary patient that we had in part one, where if a patient cuts their hand, let's say gardening, and bacteria enters that wound, what happens? Which what's going to happen next? Which type of immune response might happen? All right, so by the end of this lecture, I really want you to be able to tell me the story of an immune response. Again, if I tell you that your patient has a break in the skin and bacteria enters that tissue, you should be able to walk me through what happens next. What was supposed to keep that organism out? What responded first? Which cells show up to the party? How does the body recognize that something doesn't belong there? And eventually, how do the B and T cells get involved? So, we're going to talk about where B and T cells mature because that becomes really important as we get deeper into the adaptive immunity. So we're moving beyond just identifying cells again, and we're starting to ask what those cells are actually doing for the patient. Again, this slide groups your vocabulary into lines of defense, recognition and communication, antigen processing and response, and lymphocyte development. So here are our key terms for this section. And yes, there are a lot. But again, notice that I've grouped them instead of just giving you one gigantic vocabulary list. That first group is about defense. First, second, and third lines of defense, and innate versus adaptive immunity. Then we have the recognition and communication. So, how does an immune cell know that something is foreign? And that's where the terms like PAMPs, the PAMPs, PRR, toll-like receptors, and cytokines start becoming important. Then we move into what happens after recognition: phagocytosis, antigen presentation, MHC, complement, opsonization, and then finally we have lymphocyte development. Again, don't try to memorize all of these from this slide. I want you to just start recognizing the language. By the end of this lecture, these terms should start fitting into that larger picture. So now we meet the cast of our characters. We've already talked about several of these cells, but now we're going to organize them according to what they actually do in an immune response. And one thing I really want you to avoid is thinking neutrophil equals innate. B cell equals adaptive and stopping there. That's technically useful, but immunology is so much more interconnected than that. Some cells are primarily innate, whereas some are adaptive, and some are incredibly important because they connect the two systems together. The immune system is so beautiful, the human body, and the way that it cohesively interacts is beautiful. So here's our quick sorting exercise. On the innate side, we've got neutrophils, macrophages, dendritic cells, mast cells, and natural killer cells. And then on the adaptive side, our big players are our B cells and T cells. Um, but I want you to also look at that third category, that bridge. Dendritic cells and macrophages can encounter something as part of the innate response, process that antigen, and then present it to the cells of the adaptive immune system. So imagine a macrophage is essentially saying, "Hey, I found this. You guys need to know about it." And that's one of the ways the innate immune response helps determine what happens next. So let's compare two cells that you should already be familiar with from hematology, and that's our neutrophils and macrophages. So again, think of the neutrophil as the rapid response team. Something happens, inflammatory signals go out, and neutrophils are recruited quickly. They get in, they do their job, and they're relatively short lived. Whereas macrophages are different. Many are already living in the tissues, they're longer lived, they phagocytize, but they're also communicating. They release signals, they interact with other immune cells, and they can present antigens. So a macrophage isn't just eating bacteria, it's gathering information while it's doing it. So that's going to become really important when we talk about how innate immunity transitions into adaptive immunity. So if there's one cell that I want you to associate with the phrase bridge between innate and adaptive immunity, it's the dendritic cell. Our dendritic cells are excellent antigen presenting cells. They can encounter a pathogen out in the tissues, capture the antigen from it, process that antigen, and then essentially take that information to the T cells. So think about that sequence. Something foreign enters the body. The innate immune system recognizes it. A dendritic cell captures information about it, and now that information can be shown to the adaptive immune system. And that's how we start moving from something foreign is here to we know exactly what we're targeting. All right, so this slide's really an organizational slide. On one side, you see that we have our granulocytes, which include our neutrophils, eosinophils, basophils, and the mast cells. Then we have those mononuclear phagocyte systems, which include our monocytes, macrophages, and those dendritic cells. And then our lymphocytes, particularly the B and T cells, plus plasma cells, which we'll talk about here in a little bit. Again, you do not need to memorize any of this information word for word. What I want you to start doing though is connecting it, right? Cell, function, part of the immune response. If I gave you a neutrophil, what's its job? If I gave you a dendritic cell, why do I even care? If I gave you a plasma cell, what should immediately come to your mind? That's the level of thinking that we're really trying to build toward. So this slide might look like a lot of information, but I actually just want to simplify it into 3 jobs. Okay. So first, recognize something foreign. Second, capture and process information about it so the rest of the immune system knows what we're dealing with. And then third is to eliminate it. That's really the immune response in a nutshell. Recognize, communicate, eliminate. Different cells specialize in different parts of that process, but nobody is working alone. And that's a theme I want you to keep throughout this entire immunology class. The immune system is a network, not a collection of isolated cells. All right, now let's switch gears a little bit and talk about our adaptive cells. So here's one of those things I absolutely want you to know. Again, B cells mature in the bone marrow and T cells mature in the thymus. Again, an easy way to remember is B for bone marrow, T for thymus. But the maturation isn't just about where our cells live. These cells essentially have to go through a very specific screening process. We don't just want lymphocytes running around aggressively responding to our own tissues, right? So there's really a selection process that helps remove inappropriate or self reactive populations. Once they're mature, these cells then circulate into secondary lymphoid tissues where they can then encounter an antigen. And eventually, when a B cell is activated and differentiates, one possible outcome is a plasma cell. And what do plasma cells do? They have antibody babies. So plasma cells essentially become an antibody factory. So again, that connection, B cells then go to plasma cells then go to antibodies. So this is one that you're going to use over and over and over again, and you're going to start to see multiple courses as well. So this picture is probably one of the most useful slides in this entire lecture, honestly. Looking at the timeline along the bottom, a pathogen enters the body, and the body doesn't wait several days to do something about it. The innate response really happens immediately, right? You see here our microbe. It's passing through the epithelial barriers. So you've got barriers, you've got phagocytes, dendritic cells, natural killer cells, complement, all reacting relatively quickly. But look what happens over time, right? Now the B cells and T cells start becoming involved, and they start activating and expanding. The B cells then differentiate into those plasma cells, and those T cells then develop into effector cells. So the innate immunity buys us time. Adaptive immunity takes longer to develop, but once it does, it gives us something the innate immunity doesn't. It doesn't have the time to give us to the same degree, and that's the specificity and memory. So that's why these two systems really complement each other rather than compete with one another. So before we even need our neutrophils or antibodies, our body actually has a much simpler strategy. Don't let the pathogen in, and that is our first line of defense. So, intact skin is a barrier. Mucosal surfaces are barriers. Tears, saliva, secretions, and even the physical movement that washes organisms away are part of this defense. And then we have our normal microbiota. So we sometimes think of microorganisms as automatically being bad, but that's absolutely not true. Our normal microbiota interacts with and influences immune function. So when we talk about defense, don't just jump to white blood cells. Sometimes the best immune response is preventing the organism from even crossing the barrier in the first place, right? All right. So let's say the first line fails. Maybe our patient gets a cut, like what we talked about in the beginning of this lecture, and bacteria has now crossed the barrier. We move into the second line of defense, which is the innate immunity. This response is fast and relatively non-specific, right? That's when our cells, like our neutrophils and macrophages, are starting to respond, and inflammation's going to occur. And those soluble components, like complement, can participate. But if that organism persists, now we have to start bringing in our third line of defense. Um, and that's our adaptive immunity. So there are two words I want you to attach to adaptive immunity: specificity and memory. Okay? Adaptive immunity, I want you to automatically think specificity and memory. So it targets particular antigens, and after exposure, those memory cells can still remain. That's why the response to something you've encountered before can be very different from your very first exposure. So now look at this picture, and let's look at and tell the story from the outside in, right? We have bacteria outside of our body. What stops them first? The intact barrier. Then we have injury, a cut, for example, and that barrier's been breached. Now we're inside the tissue, and our second line of defense becomes important. Our inflammation, our macrophages, our phagocytosis, and the other innate mechanisms. And then eventually we can recruit a specific adaptive response. So, again, that's how I want you to think about these three lines of defense, not as three individual definitions for a quiz, but what happens in sequence when our patient is exposed to a pathogen. Again, first line of defense is that mechanical, mucosal. There's an injury, it's breaking down that first. Second line of defense, those cytokines get involved, those neutrophils, phagocytosis. Your innate immunity is trying its best to take care of your body, right? And get rid of that foreign invader. But then to your third line of defense, where you have the specific immune response that's happening in that adaptive immunity. All right, so now we're really starting to dive into the adaptive immunity, um, and into the different strategies. So, humoral immunity is primarily associated with our B cells and our antibodies. So, think about an extracellular organism, something that's out where an antibody can actually get to it. A B cell recognizes the appropriate antigen and becomes activated and eventually differentiates into a plasma cell. Remember that plasma cell is going to start making baby antibodies, so it's going to start pooping out a bunch of a large amount of antibodies. But then look at the other side of this diagram here, right? What if the pathogen is hiding inside one of our cells? The antibody can't simply reach inside that cell and pull the virus out, right? That's where cell mediated immunity becomes so important. So, really useful question is, where is the pathogen? Extracellular versus intracellular can really help you predict what type of immune response is going to be the most effective. So take a second and look at the comparison here. At this point, I don't want innate and adaptive immunity to feel like two lists that you're memorizing. I really want you to see the relationship. Innate immunity is there quickly and it recognizes broad signs that something is wrong. It contains the threat and it sends the signal, calls for backup. An adaptive immunity takes that information and it develops a targeted response. So instead of asking, is innate better or is adaptive better, the answer is neither, right? We need both. The effectiveness of the immune response comes from how well these systems work together. So now we need to answer a very important question. We've been saying that innate immune cells recognize pathogens, but how? They don't recognize every individual organism the way that adaptive immunity recognizes specific antigens. Instead, the innate system looks for common patterns. So these are the PAMPs or PAMPs, which are the pathogen associated molecular patterns. So I want you to think of the PAMPs or PAMPs as molecular features that are basically saying this looks microbial. And immune cells have receptors that are designed to recognize those patterns. Those are the pattern recognition receptors or PRRs. One important family of PRRs is the toll like receptors. So here's the relationship I really want you to remember. The PAMP or PAMP is the pattern, and the PRR is the receptor that recognizes that pattern. So recognition is going to then trigger a downstream response of signaling, inflammation, complement activation, opsonization, and phagocytosis. So this is how the innate immunity can respond quickly without needing to identify the exact organism first. All right, so before we finish, let's see whether we can tell the whole story without looking back. So, recall a pathogen is sitting outside of the body. What's the first line of defense? Now, let's say that pathogen breaches the skin via a cut. which part of the immune system is responding first? If you said innate system or thought it in your head, great work. So now, how does that system recognize something that doesn't belong? Remember, recognition. Now we're looking for those PAMPs, right? Which are recognized by the PRRs. What might happen next? Think inflammation, phagocytosis, complement activation, and recruitment of additional immune cells. And who can help carry out that information forward into adaptive immunity? Are antigen presenting cells, specifically those dendritic cells, right? Those are our bridge between the innate and adaptive immunity. So then we activate our lymphocytes. Remember where our T cells mature? If you said thymus, very good. And that means our B cells matured in the bone marrow. Perfect. And if a B cell becomes an antibody secreting cell, what is it called? Excuse me, a plasma cell. And then finally, what's one of the biggest adaptive immunity, biggest things that the adaptive immunity gives us after the infection is gone? If you said memory, then bingo. If you can walk yourself through each sequence and explain why each step happens, then you've got the big picture that I want you to take away from this lecture. As always, if you have any questions, comments, concerns, please do not hesitate to reach out. That is what I'm here for. I really want you guys to be successful. There's one more lecture in this series, and we're going to talk about those soluble mediators of the immune system. So I'll see you in the next recording.